It's a small typo in the notes in the page number 6. In the second line, when it says n times delta n squared minus bn, that should be a plus. It's a small sign typo. And the second one is, I don't know if you already started trying the exercises. The number 2, it's about a Lie effect. And you have to assume that A is smaller than K, when in the model equation, it's something like this. So you have to assume that A is smaller than K, okay, to solve it. You can try without this condition and see what happens, and then with it and see how things change. So this being said, let's summarize a little bit what we were saying yesterday with now showing the actual data. These are the plots that I was trying to copy in the Blackboard. If you remember, this was the growth of 2 populations of yeast that were growing separately from each other. We derived some model that was explaining how this population was growing and the interactions that were happening between cells and the resources. We got a model equation, and when you solve that equation and you find the parameters that fit the experimental result in the best way, you obtain this very nice fitting. Okay? What we're going to do today is we are going to extend this experiment to the case in which you take these 2 populations, you mix them together, and you repeat the experiment. But now, instead of having all the cells being somehow identical, let's assume, we have cells that are of 2 different types. So what would you expect that could happen if we do that? What do you think is going to happen if I measure the number of cells that I have at the end of the experiment if I start with a population that was a mix of these 2? What do I get at the end? Or what can I get at the end? Any intuition? One population goes to 0? Only that? The 2 species coexisting at the same time, that's another option. And in principle, I don't have more options, no? Or either one survives and the other one dies, or both of them. Well, or both of them die if I have like a super bad medium. Well, actually, what Gauss found in his experiment was that they both were coexisting. So the darker points, both in blue and red, is just the data I was showing before, so it's reproducing that. And the lighter ones are what he got when he was repeating the experiment with a mixed population and then measuring the number of cells that were belonging to each of the species. So he did not only find that both of them are present in the final population, you can also see, what do you see? Sorry? They grow to more slowly and to a lower population size, right? So both of them are growing less than what they would grow if they are alone. So following the way of thinking that we were using yesterday, what we are going to do today is to try to figure out both what's going on for having this joint population growth and what's happening to have this result and what would be the conditions to have other options. So what we are having here is nothing else but 2 different species that are competing for the same resource. And this is one of the possible ecological interactions. So what happens in nature when we observe so many species in the same place is that they are interacting with each other, and they interact in very different ways. Competition, that is the one that we are having here, is one of them. So if we have a species 1 and a species 2, a competitive interaction is basically that 1 is inhibiting the growth of 2, and 2 is inhibiting the growth of 2. And 2 is inhibiting the growth of 1. So do you have in mind any example that you could give me about this competitive interaction in nature? I don't know. 1. Sorry? Foxes and rabbits? Who agrees with that? Or who doesn't agree with that? Sorry? You don't agree? So do you have another example? No? So for instance, an example would be two bacteria eating the same carbon source. That would be an example. Two carnivores eating the same herbivore, for instance. Two herbivores living in the same island and having to fight for the same space. All those are examples of competition. Another example of ecological interaction is predation, prey-predator interaction. In this case, if species 1 is the predator and species 2 is the prey, species 1 inhibits the growth of 2, and 2 promotes the growth of 1. Do we have some example about the prey-predator interaction? Foxes and rabbits? Okay. Foxes and rabbits is a good example of prey-predator interaction. Another example that is the same as a prey-predator interaction is a parasite-host interaction, in which some species exploit the other species to grow and inhibit their growth. So this could be a prey-predator or a host parasite. For instance, viruses are a good example of a host-parasite interaction. And the third major interaction that we can have in nature is what is called a mutualistic interaction, in which both of them, species 1 favors the growth of 2, and 2 favors the growth of 1. One example of mutualistic interaction, for instance? Sorry? Bees and flowers are a good example of a mutualistic interaction. Another example that I found very interesting is there are some, for instance, some birds that eat the parasites that live in some mammals, and that's a mutualistic interaction between the bird and the mammal, because the bird gets food and the mammal gets rid of the parasites. And actually, that's a very good example of how, although we are going to focus only on two species interactions. Yeah. Farming? What do you mean by farming? Like if, well, you need one species farming for the other, and the other farming for. So if they both farm for each other and they both get benefit, then they are interacting. Yeah. But if one farms and the other one does not farm and only gets the food, then that's a parasite, right? So if you farm for me and I just stay at home, and whenever you come with the food, I eat all your food and I don't work, then you're favoring me, but I'm not favoring you at all. I'm removing your food, right? So it depends on the kind of farming that you have in mind. So as I was saying, we are going to focus only on two species interactions, but we can have more than two species. For instance, this case of the birds that I was talking, in that case, we have two species that are interacting on a mutualistic way, but we also have the parasites that are parasitizing the mammals, and then that leads to more than two species interactions that we can have some of them being competitive, some of them being mutualistic, and then these simple models can grow up into a much more complex models that are usually modeled using graph theory, as we were saying the hour before, and that's when biology meets quantum field theory, I guess. So anyway, the main goal that we're going to follow today is how can we put these interactions into a mathematical framework that gives us some intuition about this, and how can we analyze those mathematical models, okay? So how would you try to extend the model that we had before, we had yesterday? I'm going to use the same notation. So this is what we had before, what we had yesterday, right? Rewritten using a current capacity as we were defining. So if now we have a second species, in principle, this is describing us the dynamic, the growth of each of the species, but they are not connected so far, right? Oh, yes, sorry. Thank you. So how would we, what's the most immediate way of coupling these equations? Well, I can consider that the growth, the population growth of a species 1, is either enhanced or inhibited by the presence of the second species, and the same for the second species. Okay? And of course, the signs that I'm using in these second terms are going to give us the nature of the interaction. So positive signs mean positive interactions, and negative signs mean negative interactions. So now that we have these models for the multi-species interactions, when we extended these equations, we have something that is not very useful. That is that we have gone from having only 2 parameters, k and b, to having a bunch of parameters. In this case, now we have 6, and that's usually not very useful because we have to deal with all those parameters, and that's not the best case. So something that it's very useful to address this kind of systems is to define non-dimensional quantities in a way that we can reduce the number of parameters. And defining those non-dimensional quantities is not, there is not a unique set of non-dimensional variables and parameters that is going to give us non-dimensional equations. You can find many others, and the optimal one depends on the kind of questions that we have. Okay? So for this case, we are going to define these non-dimensional variables. So we are going to use a non-dimensional density for the population 1 that is equal to n over k1. The same for 2. We are going to define a non-dimensional time. A ratio between per capita growth rates and also non-dimensional interaction coefficients. Just to keep the notation of the notes. So if we work with these non-dimensional with these non-dimensional quantities, we can rewrite these equations in terms of these new variables and parameters, and what we get is a much more simple system. So by doing this transformation, we've reduced the number of parameters from 6 to 3. And in addition to that, we have one of them here that, as we are going to see later, is not very important for the kind of analysis that we want to do. Okay? So now we have a pair of differential equations that is describing the growth of both populations. So given this system, what would you try to do now in order to try to understand what was happening in the experiment and given what we were doing yesterday? Solving? Well, that was, first of all, trying to solve. In this case, is not as easy as yesterday. So finding the steady states would be one option. In this case, because we have non-dimensionalized this in a quite good way, finding the steady states is very easy. One of them it's basically we have three steady states that are very easy to find. The first one, which is the first one? Okay. 0, 0. Another one. 1, 0. 0, 1. And then we have a fourth one that is a non-trivial steady state that is going to depend, of course, on these signs that I fixed, and is going to depend on the interaction between the species. So because we already agreed that we were looking into a competitive interaction, we are going to fix competition as the interaction that we are going to discuss. You have one exercise that is about mutualism, that you can basically rewrite the equations and see how the whole analysis changed. But let's keep competition. So for competition, as we were saying, because this is an inhibitory interaction between the growth of the species, we need to have a negative sign there. Okay? So in order to find the non-trivial steady state, we basically need to see when these two factors vanish, are equal to 0. So if we solve so this is one, and the second one, you plug this U2 here, and you get the second fixed point, okay? So for this case, we can still, because we are keeping this interaction very simple, and our model is a very, very toy model, we can still find the fixed points. But in general, we might not be able to do that, okay? Anyway, we have these two fixed points, but sometimes they will be, they depend on the interaction coefficients, basically. This is, of course, only true if A21, A12 is not equal to 1. And in order to have this fixed point with a biological system to be like to be meaningful, we need to impose to this fixed point basically that the populations have to be positive, right? So this point is only going to be meaningful if both U1 and U2 are greater than 0. If they are not, then this fixed point is not, doesn't have any biological meaning. So in which conditions do we have that fixed point? How would you try to figure that out without, of course, equating that to 0 and solving? Do you think we could do something similar to what we were doing yesterday, or in this case, we cannot? In principle, we can, no? So the way we are going to attack that problem is we are going to draw the growth rate. Okay, we are going to grow the growth rates in a phase space, and we are going to try to see whether we can get some information about the geometry from the geometry of those growth rates, whether we can get some information about the fixed points of the system, okay? But before we go to that, one very, very small question. Why do you think that fixed point is important? Why I'm focusing so much in that fixed point? Okay, that's the only case in which I have both the species at the same time not being equal to 0. So that's the fixed point of the system that is giving me coexistence. And because I'm interested in understanding the conditions in which species may coexist, then I want to know if by studying this fixed point and studying the conditions in which this fixed point is both realistic and stable, I can learn something about the interactions between species that can drive coexistence. Okay, in any other case, I only have that either both species die or one kills the other. So if we, because we're interested again in the fixed points, we can plot in a U1, U2 space the curves in which, each of the growth rates of each of the species are equal to 0. Okay, so this basically, if I call this F1 of U1, U2, and I call this F2 of U1, U2, these two curves that are called null lines are going to give me the information about for which values of U1 and U2 the growth rate of each of the species vanishes independently from each other. Okay, so in this case, F1 and F2 are just they are straight lines, so it's going to be very, very easy to work with them, and we're going to use them to gain some practice and some intuition of how to construct this phase space. And and then of course from there you will be able to build like more complicated scenarios and gain some more practice for more complex problems. So for these two lines here, how many cases do we have? We have A12 and A21 as the control parameters. They are the free parameters, and we have 4 different cases. No? In one case we have that both of them are positive, both of them can be negative, one can be positive and the other one negative, and the other way around. No? And depending on each of these cases, then the slopes of these lines are going to be different, and we're going to have different intersections between them and the axis. So I'm going to solve only 2 cases here. The other two, is one is going to be very, very similar to one of the cases that we're going to solve here, and the other one is going to be a little more tricky, and you can do it at home. And if you don't know how to work it out, we can discuss it tomorrow. Okay. So the first one that we are going to work is the case when A12 is smaller than 1 and A12 is greater than 1. So in this case, the null lines are so one of them for U1 The first nucline is going to be U1 equal to 0. So this is basically, which one it is? So who's following me so far? Be totally fair. Okay.
I did not understand the question.
Sorry?
I did not understand the question.
So we are going to try to plot the nuclines for this first equation. Okay? So which are the nuclines? So for which, how do I get this equation equal to 0? Okay. So the first one is U1 equal to 0, right? Which one is that nucline? How would you plot this? U2. Okay. The other nucline is when the other factor is equal to 0, right? So I have 1 minus U1 minus 1A2 U2 equal to 0. Right? And we are studying the case where A12 is smaller than 1 and A21 is larger than 1. Okay? So if sorry. Thank you. No. No, it's 1, sorry. It's 1 because the idea is the coefficient that it goes with the other species has to be greater or smaller than the coefficient that goes with its own species. So then, depending on whether one is stronger than the other, is when this is going to change. Okay, it's 1, it's not 0. So we have to plot this line, and basically we can calculate intersection points. When U1 is equal to 0, then U2 is going to be equal to 1 over A12. So when U1 is equal to 0, we have a crossing point with the U2 axis, that is 1 over A12. And when U2 is equal to 0, then we have that U1 is equal to 1, okay? So what can we know about this A1 over A12? It's bigger than? Okay. So we can place the 1, let's say here. So if we repeat the same calculation for this other nucline, for this other variable, what we find is that one of the nuclines is U2 equal to 0, which is the U1 axis. And the other one is, again, another line that has a slope with crossing points in 1 and in 1 over A21 because of the symmetry of the equation. So what can we know about 1 over A21? It's smaller than 1. So it's somewhere here, no? So how can we identify the fixed points now that we have these lines drawn here? Where they intercept. So where they intercept? 0, 0? Something? Anyone else? This one?
No.
This one?
I
Yeah.
don't understand what you are asking now.
So what I'm asking is, I know that the fixed points are the points, the U1, U2 values for which these two vanish at the same time, right? So from these curves, how can I find the fixed points?
A section between the lines of a different color.
Okay. So we have those three fixed points, no? 1, 2, and 3. Assuming that U1 and U2 have to be positive, of course. If they could be negative, then I can extend those lines. But we were saying that we are interested in looking into the conditions for that non-trivial fixed point to have U1 and U2 positive, because we are looking at populations, and populations cannot be negative. So now we have the fixed points identified, and we want to, what do we want to know about these fixed points? The stability? So how can we know the stability of the fixed points given the information that we have so far? Draw arrows in the lines. So how do we draw those arrows in the lines? So I know that in this line, for instance, in this null line, I know that the arrow can only be in the direction of one of the populations, because the other one I know that is equal to 0. Okay? So let's see if they're... Okay. So here I have an arrow. Let's draw the easy part of the arrow. And now I need to know the hard part of the arrow, which is the direction. In which direction is this arrow pointing? Towards 1? Why?
Because the other population is 0, so we get to be...
Okay, that's the very good intuitive argument. I think I'm making a mess here with arrows. So the other argument is that if you look into if you substitute in the equations, then you get that this is positive. So let's go for the other easy one, this one. In which direction is the arrow pointing there? For the same reason, why? Right? Do we have any other easy one to draw? What about these two lines? In these two lines, I also know that the change in one of the species is 0, right? Because these lines, although they are not so as trivial as these ones, but they are also null lines. So here, the dynamics of one of the population is also 0. So in principle, if I'm moving around this line, the growth of the population has to be as well in the direction of the other one, right? Now the tricky question, in which direction? It depends on what? So the rate here was positive, right? The rate of growth of the population 2, or population 1, sorry, here was positive. And we know it becomes 0 when it crosses this line. right? Sorry? Still positive, right? If I don't cross 0, it's still positive. How is it here? Negative? So let's go for the final one. How is the... Okay, I'm going to, again, do the easy, and then you're going to do the hard. What's the direction of the arrow here? Negative? Are you all following me so far? Please, if not, tell me. Yes.
Explain again why the arrow there on the line is vertical.
Here? Because I know that here I have the derivative of u2 on this line is 0. Because I draw this line specifically imposing that condition. Right? So then I know that I can only go in the direction of one of the variables. So here we were saying that the direction was this one. Who doesn't agree with this? We are on the same page? Okay. And here, because I'm not crossing any null line, I have the same. This one? So here I know that this goes in this direction. Right? And I know that that over this line, the change in the direction of this variable is equal to 0. So if here it goes, if it is positive here, and I'm crossing 0, then here it is negative. Do we all see that? Who doesn't see this? Okay. So now we have in different regions of this space, we have the rate of change of the qualitative change of these variables. And from this, we can construct the stability of the fixed points. Or we can't. We can, right? Because I know that whenever I'm here, I'm going to move to this point. Whenever I'm here, I'm going to move to this point. But whenever I'm here, I'm going to move to this point. So basically, this is an stable fixed point because all the arrows are pointing to it. And this is an unstable fixed point in the sense that whenever, if I move in this axis, it's going to be stable. But if I perturb the system, I'm going to leave towards the other population, towards the other current capacity. Sorry. And what about this one? Unstable? Because all the arrows are pointing outside of it. Okay? So we know that whenever we have these conditions for the interaction between the species, we have only 3 fixed points. We have 0, 0. We have 1, 0, and 0, 1. And we know that u1 equal to 1 and u2 equal to 0 is stable. So now going back to a little bit to the biology, what is this result telling us about the interaction between the species? I know that whenever A12 is smaller than 1 and A21 is greater than 1, I have this stability in the fixed points. So in the going back to the original parameters, A12 is equal to alpha is the other way, right? So from here I know that if this is more than 1, this means that alpha 1, 2 over K1 is smaller than 1 over K2, and alpha 2, 1 over K2 is larger than 1 over K1. So if you
So the first one is not stable for a fixed point. The second one is a stable fixed point, and the third one is like a method stable one.
so this one, it's stable only is stable in this direction,
Yes.
and it's unstable in all the other directions. We're gonna do the linear stability analysis properly now, and we're gonna see what does it mean. Okay. So what are these conditions? If you look into the original equations that we wrote for the model, what are these conditions telling us about the interactions? So this term, let me write the equations again. So alpha 1, 2 over K1 is this coefficient over here, right? And 1 over sorry?
I think yeah, this is right.
What is right?
This makes sense.
Why does it make sense?
Because the presence of N2 they will have two effects. Not more than two, but among those make N1 decreases and N2 decreases. And we this tells us that the presence of N2 is worse for the N2. So anyone prevalence.
Do we all agree? So this is telling us that the way that the species 2 inhibits the growth of a species 1 is weaker than the way it inhibits its own growth. So the competition between 2 and 2 is stronger than between 2 and 1. The 1, 2 competes against 1 is weaker than the way it competes against itself. And because it does that, then it's more harmful for its own growth, and therefore species 1 is going to outcompete it, because in the case of a species 1, we have the opposite relationship. Okay. We all see that? So let's do the interesting case, the most interesting one. So again, the other three cases are, they are also useful and they're interesting. And I recommend you to work them, but to be able to cover the whole material, let's jump to let's jump to the case where A12 is smaller than 1 and A21 is smaller than 1. Okay? And let's do it big so there are no messes. So again, we have these null clients and we're going to plot them again. We're going to go a little bit faster with the trivial ones. So the first one is U1 equal to 0. U1 equal to 0 is the U2 axis. We have U2 equal to 0. And then we have the other ones that we had that come from the other factor. Okay, so now remember that we have A12 and A21 smaller than 1. So therefore, the inverse are always larger than 1. So this is 0. This is 1. This is 1. And this is 1 over so this comes from A to 1, right? And this is 1 over 1 over A12. Okay. So now notice that because we have the different slopes, we have the non-trivial steady state for positive populations. So now the fixed points that we have are 1, 2, 3, and 4. If we now draw the same arrows, how would they go? So how are the arrows going in this null line? Towards 1. Here, towards 1. Here. This way, because I'm not crossing the null line yet. Here, this way. Here. Down. Here. That was too fast. And here. So we can already see that all the arrows for this fixed point, whenever I approach it, the arrows point in the direction of this point, which is telling us that this is the stability of this point, and this is an unstable fixed point now. Okay? And if we work with the coefficients, as we were doing before, well, yeah. So this is 1 and this is 2. Is it? No. So now, because these two coefficients are smaller than 1, this means that alpha 1, 2 over k1 is smaller than 1 over k2. That if we go back to the equations, it's telling us something different than what we had before. What it's telling us is that both species are competing with themselves more stronger than with the other species, and therefore they allow the other one to survive. But there is something else that we can learn about this analysis, not only that we have a species that may coexist because the competition with the other is weaker than with themselves. It also that, as you can see, the fixed point here, although in principle, we don't know the value of the population for this fixed point, we do know that it's below the population size that we would get if populations were growing alone. So because of the presence of the other species, the populations interact with the other and they grow to a population size that is smaller than what they get when they are alone. And this is the result that we were trying to explain. So one hypothesis to explain that result is that because these two species of yeast are interacting with each other more strongly than what they are interacting with the competitor, then they reach an equilibrium in which both of them can coexist. Okay, and this is, of course, the most basic mechanism by which a species may coexist. There are many others. When you go out there, you have seasons, you have a spatial structure, and you have these interactions to be made much, much more complicated. This is just one of the possible ways in which these species may coexist. So this is a very useful way of analyzing these kind of problems, but of course, it's not the ideal one because you have to draw these portraits, and then you need to gain some intuition, and sometimes you may not get all the information. And it's always convenient to be able to have some actual stability analysis of these systems. And actually, what we were doing yesterday for 1 dimension can be extended to 2 dimensions, and you can obtain the stability of these fixed points on a more systematic way. Okay? So to finish, we are going to see very quickly how you can do that. It's a very useful way to get the stability of the fixed points, and it's going to allow us to classify the stability of these fixed points on a very, very systematic way. So in the more general case, we'll have dynamical system that can be written in this way, no? And following what we were doing yesterday, we know, what we want to know is how this system is going to respond against perturbations. So we can write okay? And if we insert this here, and we do the Taylor expansion for each of the equations that we had before, that we did yesterday, okay? I'm just doing the Taylor expansion of a function with two variables. And I can do the same for variable 2, whatever. And now this, of course, as we were saying yesterday, this is equal to 0, the same in this term here. Yeah, I was. And now I can write this equation using matrices. If I define a vector C where A where A is the Jacobian, is the matrix that you construct with the derivative of the function. So now for this equation, I know that this, in principle, the solution for this equation is going to be exponential, right? So I can write I can hypothesize, use these ansatz as to solve this equation. And at the end, what I obtain, I can reduce this to an eigenvalue problem, basically. Right? I should, so let's do. So how many of you know this? All right. So then at the end, you get that basically the solution is a combination of exponentials in which the rate of each exponential are going to be the eigenvalues of this matrix. Okay? So basically, if you're able to obtain the eigenvalues of the Jacobian of your dynamical system, you can know what's the sign of the growth of the perturbations. So if both eigenvalues are positive, this means that your solutions are going to grow in time and your fixed points are going to be unstable. If they are negative, they are going to decay in time and your fixed point is stable. If they have different signs, what does it mean? If I have a fixed point and the perturbation in one direction is positive, has a positive growth rate, and in the other direction has a negative growth rate, so this is called a saddle point. It means that depending on the direction of the perturbation, your fixed point is either stable or unstable. And there is another possibility, which is that your eigenvalues of A are complex. So if you have complex eigenvalues, what are the possibilities that you may have? If the real part is 0, then your solution is basically going to be a linear composition of sinus and cosine. And then you have oscillations, so you have your fixed point, and you have oscillations around it, or they may have a real part that is positive or negative, and if the real part is positive, you have oscillations that decay, and if your real part is positive, you have oscillations that explode. So very briefly, what I wanted to give you is an idea that there is also an analytical way, well, analytical, at least a systematic way of doing this, because of course, depending on how many equations you have and depending on how they are, you will have a very large matrix that you may need to linearize numerically, or you may not be able to obtain the eigenvalues analytically, but you can do this kind of stability analysis that we were doing before on a systematic way and get the stability of the points depending on the conditions that you are studying. So this is what I wanted to show you today. Tomorrow we are going to move to the case where we relax this assumption of individuals being well mixed. We are going to have a spatial structure in the population, and we are going to see how the equations change and how the solutions change. And there is an exercise about this part in the web page, which is basically considering the mutualistic case and going through all these calculations. And I don't know if you have any question or comment. No? Okay. So then I will stop here and I will see you tomorrow.